Hi everyone, my name is Glib and I'll be talking today about my research proposal for my master's work. The topic of my thesis is development of a high-throughput screening approach to identify production enhancers of adeno-associated virus, or AAV. AAV is used in gene therapy, and as we all know, that market is huge. As of 2020, there were 32 gene therapy clinical trials that were approved worldwide, and 70% are viral vector-based. The gene therapy market is huge with $3 billion currently, and it's expected to be $16 billion by 2030. AAV holds a questionable title for being the most expensive drug on the market and in history ever, with its $2.1 million per dose, which is outrageously expensive. But one of the reasons why it's so expensive is it's really difficult to produce high titer and high quality AAV. The goal of my project is to establish a screening platform that will allow to do screen looking for chemicals that help boost production of AAV, hence decreasing the price of the gene therapies. So quickly looking at the AAV, what it looks like, it's a replication deficient single-stranded DNA virus with a 4.7 kb genome. It has two open reading frames, RAP encoding for replication proteins and CAP for capsid proteins, and two ITRs, which are inverted terminal repeats on each side. The most common way of production of AAV is triple transfection method. So you have three plasmids. First one is your transgene plasmid, where you basically put the transgene of interest. Second one is the RepCap, so it's the plasmid that encodes for the replication proteins that will help to make AAV, and the capsid proteins that for the outside shell of the AAV virus. And the last one is the P helper. So this plasmid mimics the co-infection with a helper virus, which is necessary for AAV production. There are a whole bunch of AAV serotypes. There are hundreds naturally occurring, but the most commonly used ones are AAV1 to AAV11. It's really important to point out that in my master's work, I'll be using only AAV2 and AAV8. AAV2 is the best one out of all of them in vitro. It transduces really well. AAV8 is the worst one out of all of those in vitro, but it's the best in vivo. So that's why it's a topic of interest. Super quickly looking at the cellular pathways of AAV. So how it gets in the cell and how it gets replicated. So firstly, it attaches to the membrane, gets in the cell, translocates to the nucleus, uncoats, expresses its genes, replicates itself, and assembles the complete virions. It's important to point out that unlike other viruses, for example, VSV, vascular stomatitis virus, or herpes virus, or Marburg virus, the MOI of those viruses needed is very low. So it's about 0.01 or something in that category. For the AAV, the MOI, so the multiplicity of infection, or how many viral particles per cell you need to get the transduction going, it's at least 1,000, which is a lot. So that's one of the roadblocks. Now, there was some research done looking at these receptor proteins. They're called AAV receptor proteins, and it was found that cells that express this receptor protein, the MOI needed for the AAV to transduce them goes to as low as 0.1, which is very interesting, and it's something that I'll be exploring in my work. Okay, so I mentioned the production enhancers, and so what is this? Well, there was work done performed by Dr. Diallo here at the OHRI. He screened chemical libraries looking for production enhancers for the VSV virus. And he found a whole bunch of chemicals that increase the production of VSV, and one of them boosts the production up to 35-fold, which is huge. And if there's something for VSV, maybe there's going to be something for AAV. One way to find out. With that in mind, let's look at the aims and hypotheses. Aim number one is to develop a high-throughput method of detection and quantification of AAV. And aim number two is to develop cellular systems that will allow high sensitivity of high-throughput detection. With them combined, we hypothesize that if we use the cell line that expresses AAVR, that receptor protein that I just talked about, in combination with the optimized parameters for the high-throughput screening, that will allow to establish a method for quick, accurate, and reliable way of detection of the AAV production enhancers. So let's look at the actual data. Aim number one, to develop a high throughput method of detection and quantification of AAV. Here, this step is basically done. So I have finished optimizing the high throughput screening layout for AAV2. This required quite a bit of work. Currently, to finish aim number one, I had to do 72 experiments total, and there's no time for it today, but I'll be just presenting the general idea. I had to do a whole bunch of cloning and a whole bunch of molecular biology stuff to get all of the plasmids that I wanted and make sure they're right. Start producing AAV in flasks, then scale it down to 96 plate, and then do a whole bunch of optimization to get the screen going. So the way it looks is we have three plasmids that triple transfection that I talked about. We transfect the producer cells that are seated 96 plate with these three plasmids. We treat them with the drug library, lyse the cells, transduce the reporter cells, and quantify the transgene signal. So this sounds pretty straightforward in the paper, but there are 7 main things that I had to figure out. These are main things, and there are countless other small things that have to be optimized. First one is the optimal cell producer. So which cell gives us the best amount of AAV? It was HEC293. When's the best time to treat with the drugs? You can do co-treatment, you can do pre-treatment, or post-treatment, and turns out the co-treatment was the best. The optimal transgene. So we'll look at the transgene that's optimal for the plate variability and percent CV and stuff like that. When's the best time to harvest? What's the best way to harvest? How do we go about the dilution of our crude produced and harvested AAV in the transduction? What's the best reporter cell line, and when do we read our transduction? So all of these things got optimized. Now I'm looking at the AAV8 screening layout, and something that's important to point out here that need to be optimized are the transfection ratio of the plasmid. So maybe we'll be able to, if we select a certain ratio of plasmids, maybe we'll be able to boost the production. Hence, will make things easier on the transduction side of things. And this is where the AAVR cell line comes into play. So having the AAVR cell line, in theory, should really help us out with the transduction and hence detection of our AAV8. So let's look at the aim number 2. So the aim number 2 is to basically make that AAVR cell line. So how do we do it? Well, generated the plasmid that has the AAVR in it and the neomycin resistance gene. So we'll be able to screen for the cells that have resistance to neomycin, transfect the cells, screen, so add a whole concentration of antibiotic and see the ones that survive, confirming that they have the plasmid, and then do flow cytometry. So this sounds, again, simple, but it's not as straightforward. And this gave me a little bit of headache here. Cells got transfected. I selected them with the antibiotics and then did immunofluorescence to confirm that, well, we have AAVR. And they were HeLa and HT1080. Here we're looking at the immunofluorescence. So here they got treated only with the primary antibodies, and we can see only the cell nucleus, which is great. Secondary antibodies, we also can see the nucleus. But when we're looking at the primary and secondary antibody, we see this these beautiful pictures showing us cells that have the AAVR in them. This is nice, but the problem is AAVR, R stands for the receptor protein. It's supposed to be in the surface, and it seems like it's in the cytoplasm here. Turns out that the plasmid got, there was something wrong with the sequence, and there was the secretion peptide. It was basically in the wrong place, and the receptor didn't go, the protein didn't go on the surface, so it was free floating in the cytoplasm. Quick experiment here, I just transduced these cells so that are expressing and not expressing the AAVR with the AAV to GFP just to check if we have any enhancement of the production. And I transduced with the AAV to GFP, and I was looking for the GFP counts, and we can see that not at the all of the dilutions, there was the same amount of ours, of the signal. So confirming that even though AAVR is there, it's not really boosting our transduction. So once we get the AAVR done, we might or might not need it for the AAV8 screen, because if everything else is optimized, we might not require the AVR. Then we'll be able to perform a high throughput screen looking for the production enhancers of the AAV, and hopefully we'll be able to bring down that cost associated with the AAV gene therapies down. Thank you.